All right, so I'm going to begin with the overview of contributing data to Facebase. Okay, so the first thing we want to communicate is just that Facebase can be a great home for your data. So as you may have seen already or know from the past, we have a broad collection of data. We span a couple, primarily a couple animal models and then human subjects data. And unlike some, say, experiment-specific data repositories, we span not only the omics data, but imaging data, even reference data, and a variety of different types of data for different experiment types that fall outside of kind of the usual imaging and omics. But from all of that, there's of course a common theme that Facebase is about craniofacial development and dysmorphology, and we're also expanding to include more data on more dental data. We are interested in data on additional animal models. So we have a couple listed on our priorities, which I'll browse later. But we've talked with researchers who are developing different types of animal models, some canine, swine, and other models, and we're interested in that as well. So although Facebase has this broad collection of data, it is focused on this craniofacial development, dental data, craniofacial dysmorphology. So there's a common theme that ties your data together, and you can have both omics data as well as imaging data, which are often the case in many overall investigations. So whether you use Facebase or not, your data are first-class research products. So increasingly, the overall community and publishers are certainly helping to make this push to recognize data as a first-class product of an outcome of your research. So Facebase gives a home to that as well. So you've seen before that you have an archive quality repository, you have identifiers, we have we follow best practices for data citation. We give guides to those who use data to follow those citation practices. So we provide an overall home to make your data not only available, but with instructions for others to provide academic attribution, in making proper references to your data. And then if all else fails, of course, there are data sharing requirements, both from funding agencies as well as increasingly the publishers as well. And so sharing data through Facebase, if for no other reason, at least satisfies those requirements. But we think there are a lot of benefits. And the reason those requirements exist is because there's a lot of buy-in from the funders and the publishers, and increasingly many researchers to understand that research data are a high quality product of the overall way that we communicate a scientific result. So the first step in the process, and I'll walk through this on the site so you know where all of these links are, but the first part of the process is to tell us about your data. So there's a little form on the site just to kick off the process there, where you fill in basically some contact information for yourself, as well as a title and short summary or abstract of the data and maybe the investigation that produced the data. We do ask for details about, say, whether it's human subjects, identifiable data versus animal model data, the types of data. And then we ask for you to give a few sentences, paragraph or so, to say how you think it fits in Facebase. So again, we're very broad. There's a lot of room for different types of data, but as long as it fits in the overall theme. And we give some guidance too about our priorities, which I'll look at. But even if it doesn't fit in those priorities, we would encourage you to consider filling out the form, letting us know about it. And really, we're not overly restrictive about the fit of the data. The main issues with, say, data qualifying in Facebase that have come up have been around, say, identifiable human subject data that wasn't collected under an IRB at an institute recognized by the NIH. So things like that. So, that can be an issue, or where consenting, informed consent may not have occurred, in which case, the data can't be shared through our site, or really any others that are NIH supported in the US. So when you do fill out this form, our steering committee, which is comprised of members of the hub and the program office, program staff, reviews it, and we try to get back within a couple weeks. So if you haven't heard from us in a couple weeks, just give us a ping, and then send us a little email to see to check on us. But we generally try to get back. And then we're like this little kitty down in the corner. That's about how happy you make all of us when you fill out that form. We are very enthusiastic and encouraged to hear about your data set that you can contribute. So we really welcome any any kind of contribution that you might be able to make, and appreciate it. So I just want to repeat here, just revisit this slide about the organization of data, because not only does it affect how you navigate data in Facebase, but about the kind of data that you contribute. So just in case anyone wasn't around for the morning session, I'll just kind of repeat the parts of this. So starting from the top, and this is part of the overall contribution process of the data that you share. So there's a description of the project, who are the members of the project, the PIs of the project, the funding mechanism, funding support for the project. So that's the lab, the investigator, maybe a collaboration, could be a multi-PI type of award, and then projects contribute data sets. So data sets are just collections of data and metadata. And again, metadata are data that describe other data. And then the kind of details that we ask for to describe your data are experiments. And again, these are the particular procedures, like either an imaging assay, or RNA-seq or ChIP-seq or single cell, or other types of assays. Obviously, those will follow protocols. And increasingly, we ask for sufficient detail to go into those protocols, although they are free-form descriptions. We'll give some guidance on what you might put in those. And then, of course, experiments are performed on biosamples, and those describe the biological characteristics of the specimens that are used in the experiments. And then again, this little part that can be a bit confusing that we're working on improving, but this issue of once you describe your experiment and your biosample, then you associate the two in the form of what we call a replicate. So that's really just an association between the experiment and the biosample. So if you have 3 biosamples that are used as the controls in an imaging experiment, then those would become the 3 bioreplicates of that experiment. And you just enter the details first, and then you link them together. You identify what are the groups of replicates. So here's your control group of replicates, here's your homozygous group of mutant replicates, and so on. And then when you submit data, that data are either uploaded directly to each replicate, or they are uploaded through a bulk upload tool that as long as you use the identifiers and put them into a little directory structure, it'll just suck up all the data that you put in this little organization on your file system and upload it to Facebase and link it up to the metadata that you've entered. And again, I would just mention that the data that we that we seek are data that are suitable for reuse, reproducing of results, and analysis. So again, things like, let's say, a multi-panel figure that shows like a comparison of things between, say, control and mutant, or control heterozygous, homozygous mutants. That may be a really good part of a presentation in a paper, but and we would welcome that in Facebase. But in terms of data that's suitable for reuse, reproducing results, and so forth, what we want are like the high resolution microscopy image, the reconstructed CT data, preferably, say, the raw sequence reads, FASTQ files and such. So that's our preference, to have the higher resolution, higher fidelity data in Facebase. So just once you get approved to submit data, then the process looks something about like this. On the left-hand side, you begin with generally you begin with a tutorial. And actually, since you're here for this boot camp, you've kind of checked that box in a way, although we welcome you to do another one-on-one, and we can kind of tailor it to your specific data set too, and kind of point out some of the things that might help you with submitting your data set. So we're more than happy to do another one-on-one session. Then next up, you use online forms, which Alejandro will be showing you these tools. So you use online forms to describe the data sets, the experiments, the biosamples, and these have convenient mechanisms in them so that you can copy over rows or cells out of the as you enter data, so you don't have to like punch in each biosample. Let's say you have 40 to enter. You don't have to punch in each one, because most of those are groups of replicates actually. So you enter in one, copy it over 3 times. So it has mechanisms like that to ease the entry of data. Then the next step is you upload your data files. Again, that can be done directly on the website and upload it through your browser. But if you're dealing with, say, a gigabyte file or 10 gigabyte file, or you have a lot of files, you probably don't want to do that through the browser. So our desktop apps, which work on Windows, Mac, and Linux, they can manage a whole bulk upload of a lot of data. They do things like retrying. So if something fails, it'll restart it automatically. It'll pick up where it left off. If you rerun it at a later point over the same data set, it'll skip the ones it already has. And the way it skips is it actually computes what's called a checksum, which is a mathematical digest of the file into a small encoded form that allows, it's like a fingerprint. It's a fingerprint for your data, so you know exactly that the data is the same on the client side, the server side. So this tool does all of those steps. So it makes sure not only for handling errors and retry, but it also makes sure that the data fingerprint matches this checksum on the client side and the server side. And then there's a quality control review step. So we have some of this feedback built into the site, but we also work with you too. And again, we're more than happy to help review things and even help adjust or complete or fix something. But we work with you in this step to get the data set up to the level of quality that we want to have in Facebase. And then you'll notice that before I even get to release here, I mentioned citing data sets. So a lot of times, if you're submitting a data set, you're doing so maybe while preparing a manuscript. So we like you to think about citing at this time and actually citing your own data set, so that you're not citing us. This is not us getting a citation. This is you citing your own data set and maybe even specific parts and images of your data. And this increases the reproducibility and verifiability of the results that you report in your paper, increases the transparency of your results, where people know, okay, he's showing this image, I can go there. She's showing this diagram, I can go there. You know, that whatever you're reporting, they can find exactly the data that support a particular result or that you used as input. And then citing your own data set lets other people who encounter your paper know where to go and find that data on Facebase. And it also gives them an example of how they should cite your data in the future. And then finally, the release step. Once we're happy with the quality of the data set description and you're ready and you're happy with it, then we release it and make it public. So up to this point, no one but you, your team, members of your project can actually see your data set and all the parts of the data set until we release it. And this can also be useful if, say, you do have a manuscript in preparation and you want to embargo the data set. You're saying, well, it's going to take about 6 months to get this out, but I want to upload the data, have it all ready to go. And increasingly, that's part of the publication review process. So you want to have it all, or just part of the requirements. So you want to have it all ready to go, but you want to embargo it to a certain point. So we support that. So you can have it all ready all the way down to the last step, and then just say, okay, I want this released in a month or 2 months or so. Okay, so again, so on this topic of citing data sets and publications, so this is a little blow up of that share and cite window that comes up when you click the link in the upper right hand side of any data set. And what you see is that we have first of all, a versioned link. And this takes you to this exact view of your data set, even if things change in the future. So this is one way. If you want to share something very specific and versioned at a particular point in time, you can do that. Then there's the live link, which is useful if, say, you're sharing with someone while you're preparing the manuscript and you want to point them to exactly the data set. They would have to be in your team if it's not released, but just the latest view of it. And then finally, the data citation. So this gives an example that follows the best practices that have been outlined in the community. So you have up front the contributors, who are essentially like the authors, and you can add to that list whoever is involved in the data set. And then there's a title. Not all guidelines suggest or require titles. Some do, some don't, because data sets don't always have titles. In Facebase, we have titles as a regular required field, so you would have that there. And then the repository, again, that's a standard part of a data set citation, is you cite you include the repository that it's located at. And then we generate the digital object identifier. So you include a persistent identifier, and then finally the year of data set release. And then at the bottom there is something called BibTeX. And I don't know if everyone's familiar with that, but it's a fairly broadly supported format for that can be imported into most reference managers. So say, for instance, EndNote can import it. And if you use the free open source JabRef, that's actually, I believe BibTeX is the native underlying format for JabRef. So in other reference managers that you might use. Okay, and then just again, to revisit what a data set looks like when it's been uploaded to Facebase. So again, we have those digital object identifier at the top and the other identifiers. Attribution then comes next. And then I want to mention this cross-reference. So in some cases, you might say upload sequencing data to Geo, or human data to dbGaP, or you might have other repositories that you share data with. So you can have a variable number of related links off to other sites. So you could have links to Geo or dbGaP or others that I haven't mentioned. Basically, you can have a link to say, here's the rest of this data set. And sometimes we have that because, say, someone deposits the imaging data with Facebase, the omics data with Geo, or maybe not imaging, but they upload a gene annotation tracks, which can be viewed and annotated, visualized online through the UCSC genome browser, because we run a registered track hub for UCSC. And then they put the sequence data, actual raw data, over at Geo or something. So that's fine. There's a way to do those cross references on the Facebase site. So if that's of interest, you're able to do that. And then next up, we have, again, visualization capabilities. So as you're uploading data, consider making use of that. We're making it easier to have your data visualized online. Again, as I mentioned, we've added new pipelines. So we have a pipeline now that will take most formats of imaging data, generate thumbnails, and generate the high resolution as you give us, efficient online pan and scan viewable image visualization online. And then as well, I showed an example in the morning session, not only of that viewer, but also one that was, say, a multi-series TIFF image. So our viewer will let you step through all of them. You can do some online adjustment of just some basic image manipulation online. There's also a feature where you can annotate in there. So if you're the contributor of the data set, you could actually do your annotations online. So our pipeline will run, and say about overnight, you'll have the image processed, and you could do that online, and basically mark up and annotate with, say, some anatomical markings, some comments about it. And you could actually even take your screenshots out of that and use those as the base images for putting together those image panels that you typically might put into a publication. So you could actually prepare all of that on our site and then take screenshots and then put them together in your own collage of images to go into your publication. And then you would have very specific, when somebody sees that image set, they could actually go and see the underlying data with your annotations all live and viewable on the site. So it's a really powerful capability. And if you have high resolution data and you're thinking about annotating them, please consider using this. And if you're unclear about anything, again, we'll do a one-on-one and show you exactly how to use that. We also have, as I've mentioned before, the genome browser. So if you upload tracks, those will be visualized online. And when somebody goes directly to UCSC, they'll find your tracks in the Facebase hub. They have to activate the Facebase track hub, just like any public track hub on the UCSC site. We are working on a single cell processing pipeline. So using the UCSC tool, we're also looking into some other pipeline tools, but we're further ahead with the UCSC cell browser. So we are gearing up for making that available and automating that to the extent possible. And then we also, if you've created like segmented mesh images, surface models, you can upload those to Facebase as well, and those can be visualized right on your site. And then finally, image volumes, say TIFF or Nifty are the two primary formats that we support for this orthogonal slice viewer. You can upload data to be viewed in that online as well. Okay, so just a final word before I do a brief tour or get us started and hand over to the next presenters. So we think that there are a few strategies for success here for data contribution, and by success we really mean like lowering the effort on your side, making it easier, a more streamlined process. So first up is reviewing the guidelines early. So in a lot of ways, you've done a lot of that just by being here, but we will show you where the online documentation is, and that can help just, again, thinking about that experiments and biosamples and the protocol details that you need to upload. Just being aware of that early in your process, even while you're planning experiments or planning your data acquisition activities. It's all data that you're going to have anyways, but it's kind of like, it's helpful just to think about how to keep that organized, both for your own practices, but then it'll make it easier for uploading contributing the data to Facebase or really any other site. And then secondly, again, take the 1 hour tutorial with us before. Again, you're kind of ahead of the curve by being here, but it might be 6 months before you have something ready to upload. And if that's the case, you might want to strongly consider doing a little one-on-one tutorial with us again, or catching the next webinar that we hold down the road, because that can really help with going through and knowing where to put things, where to upload things on the site. And then third thing, you could even submit, say, one data set or even a part of a data set and just ask one of us for feedback on it. We're happy to give you that. And that can really help you avoid any systemic errors in the process. And then fourth, just as I've mentioned, we're happy to help. So just reach out to us anytime. When you're actually in this curation process, your main point of contact is Alejandro. and you may be directly contacting him, but we always encourage you to copy the help list, because Alejandro unfortunately does take vacations once in a while, and other things in life outside of Facebase. And so it's good to just be able to reach out to the rest of us anytime, and we can back him up if you need something. Okay, so with that, let me just pull up our Facebase site here. Okay, so now when you're coming to the site to submit data, you'll notice up front we've got we've got a couple links into the process. So on the nav bar from anywhere on the site, you always have this submit. So under here, you have how to submit data, you have current data priorities. Also on the home page, front and almost center, you have how to submit data to Facebase. This is the same link as how to submit data to Facebase. So both places here. Okay, so this tells you a couple things. First of all, it asks you to review our data access policies. This is mostly if you have if you have human data of any kind, you should read this. Okay, because we're a little more specific, and our guidelines in running Facebase are that we are to be a little more conservative sometimes than perhaps even your own, if you're operating in the US, your IRB determination, where you may think, oh, this is human data, but it's not identifiable, it won't be human subject data. And we may actually have to tread a little more lightly, conservatively on that data. So take a look at the data access policies. And again, this is mainly if you have any kind of human data. And then the next is the current data priorities. So this is useful to see. I wouldn't, whoops, hold on one second here. Okay, I wouldn't be too concerned about if you don't see your type of data, your investigation mentioned here. So again, we welcome others, but these are kind of the ones that are maybe our main data recruiting priorities. So sometimes we will reach out to people after a conference or a poster session or something like that. So these are the things that we're kind of have high on our agenda, but again, we welcome others. Even it's not mentioned here, we have spoken to others who have come to us with different animal models that they're developing, and we've had very enthusiastic response about that. So we're very interested. We do mention some specific gaps in our data that our advisory committee have mentioned, and then new types of data that we're exploring. Okay, let me back up now. And then we're back here on the how to submit page again. Next up, fill out the form. Okay, so this is our form that brings great joy to us when you fill it out. So again, it gives you a little reminder of what kind of details we'd like. I would say, again, the sticky issue when being able to take data is really this human subjects data and whether there's informed consent and how we can review that. We welcome it, absolutely, but that's where we have to scrutinize a little more closely. And then next up, so once you filled out the form, again, we get back in about targeting about a couple weeks if we can. And we'll usually be touching base with you about what to do next, but I'll show you where the link is here, where we'll always have many reminders and helpful communication about it. But we have our online instruction form over here, not form, but instructions over here. And I'm gonna let up here in a second as we go into the hands-on version of this. But basically, Alejandro is going to walk us through a live demonstration after Vu shows some examples of protocol documents. But this gives an outline of the overall process and all the steps, and it has screenshots of different parts in the process. And you'll kind of see, okay, there's key concepts here. Then there's the data submission process, which gives an overview. And then you'll see, create a data set, create experiment biosample replicate entries, upload data. So again, those are all parts of those key parts of the data organization that we have in Facebase. So let's just look at the key concepts. Now, here's this diagram again, giving you an overview, always reminding you of what the overall structure is. And then we have a description, as I've talked about this already. We have a description of the project data set and so on. We do talk about like our use of vocabulary. So Facebase has used vocabulary that have been developed by other researchers, informaticists in the overall bioinformatics community, or informatics community, I should say. And so where possible, we have, and available, we have adopted those vocabulary. And the benefit there is that when you're describing something in Facebase, you're usually not using just a free-form term or just describing it how you wish. You are picking from a term that has been curated by the community, and there's an official accession number, there's a preferred name, there are synonyms for it, there's a description, and other people, say, in contributing data to other repositories, can also, potentially be using those terms or related terms or synonyms of those terms. And then we can know what we're all talking about. So a lot of times our forms are very drop down heavy, because you're really just picking terms to describe that have already been agreed on by the community. If you don't see them, if you don't see an appropriate term that you want, we can go out and look for it, or you could suggest it, and we have links to the NCBO bioportal. That's a great place to look up terms. So, but we are driven by this to improve the interoperability of data. And then we talk here about specific data types and formats. And so what are we looking for in terms of sequencing data? What do we mean by process data? Well, this is like align data and track data, and then track data. Array, not as much of that, but microarray data. What do we look for in terms of imaging data? And the only update I would say, we actually are able to take a lot of vendor proprietary formats now, and we'll provide some more guidance here with some additional links. Surface models, so if you do have segmented data, we mentioned the format. OBJ is what we are able to support for online viewing. You could submit others, but that's what we can support online. Okay, and then we go through some additional sections before dropping you into the next step, which is data submission process.